Alright, so in this video, I'll be talking about Hershey and Chase experiment. Hershey and Chase experiment was carried out in 1952, where they tried to prove DNA as the what? As a substance for what? Inheritance. So Hershey and Chase used what? The T2 bacteriophage. When they used the T2 bacteriophage, they now improvise with the word the radioactive isotopes. In these radioactive isotopes, they use the words phosphorus and the sulfur. The radioactive phosphorus and radioactive what? Sulfur. So this was what happened. They used the T2 bacteriophage, which all of us, or most of us, know what is bacteriophage. It's just like it's the viruses that infect what? Bacteria. So in carrying out these experiments, Hershey and Chase used the T2 bacteriophage, where they cultured these T2 bacteriophage in two medium. So they take, for instance, this is the two medium. So they cultured this T2 bacteriophage in two medium, and in this two medium, they were containing what? Radioactive isotopes, which was phosphorus and what? Sulfur. That's radioactive phosphorus and radioactive what? Sulfur. That's to tell you that this medium was containing what? Radioactive what? Radioactive phosphorus and radioactive what? Sulfur. Same as this medium as well, it was containing radioactive phosphorus and radioactive sulfur. This medium here was containing radioactive phosphorus and radioactive sulfur. This one too was containing radioactive phosphorus and radioactive what? Sulfur. But T2, take note that in this medium, there was presence of what? T2 what? Bacteriophage. T2 bacteriophage and T2 bacteriophage in this one as well. So there was T2 bacteriophage here and T2 bacteriophage, but there were presence of what? Radioactive phosphorus and radioactive sulfur in the medium. That's to tell you that there were how many components. This T2 bacteriophage was present, radioactive phosphorus was present, radioactive sulfur was present, same to this medium as well. So when they cultured this medium, you should know that what? In DNA, there's presence of what? Phosphorus. In DNA, there's presence of phosphorus. And in what? In amino acid, because most of the amino acids, I think 16 has what? A radio sulfur on the side chain. 16 has what? Sulfur on the side chain. And most of the amino acids, they have what? Sulfur. And when it comes to what? To DNA has phosphorus at the phosphate group. So they now what they did was they now allow this medium that they have what prepared. They now allowed this medium to infect a bacterium, which was what? Escherichia coli. They now allow this is a bacteriophage. They now allow this bacteriophage that they have cultured in the medium, which they cultured in the presence of what? Phosphorus and what? Sulfur. Radioactive phosphorus and radioactive sulfur. They now allow this to take, for instance, this is the what? This is the bacteriophage. This is the bacteriophage. This is the bacteriophage, and this is the what? And this is the bacterium. Which is what? Escherichia coli. They now allowed this bacteriophage to what? To infect the bacteria, this bacterium, which is what? E. coli. So in this infecting of this bacterium, there was what? This DNA, this genetic material here, this material which was not known by then. So it was able to transform or transfer into what? To enter into the cell of this E. coli. This material inside of what? The bacteriophage was able to what? To enter the bacteria, the bacterium rather. So it infected the bacterium. So it made what? If you know that if it infects, it will make multiple copies of what? Bacteriophage. Multiple copies of bacteriophage. It will make multiple copies of bacteriophage. But at this point, they didn't allow it to make multiple copies. What they did is that they now blended this bacteriophage that infected the bacterium. As I earlier said, they allowed this bacteriophage to infect a bacteria. So they now blended what this what most times this experiment is called the blender experiment, because there was a presence, there were a use of blender in the process of the experiment. So they now used a blender. They now used a blender and blended this structure you are seeing on the board here. So when they now blended, everything was now mixed up. Everything was now mixed up. So if you blend this, when they now blended, they now was this centrifuge after the blending process. They now centrifuge. They now was centrifuge. The centrifuge after blending. So when they centrifuge after blending, they now decant. Sorry, they now separated the supernatant from the pellet, which is the residue. So when they separated the supernatant from the pellet, now observed the supernatant. When they observed the supernatant, they discovered that they saw what? Radioactive sulfur in the supernatant. Because if you know this, if you know viruses are lighter than what? Are lighter than bacteria. So the viruses were able to what? To stand up. They were that's the if you, in this situation, the supernatant was the viruses. The bacteriophage that was what? Was able to infect that bacteria. You get me? So in the bacteria there was what? The E. coli there was the bacteria, and it was the what? Pellet. It was the residue, the one that settled under, and the upper one was the what? The virus, which was the bacteriophage that was able to infect that bacteria. So they now discovered that there was what? Radioactive sulfur in the supernatant. So they now check the pellet as well. They now discovered that in the pellet there was what? There was radioactive phosphorus. There was radioactive phosphorus in the pellet, and there was what? Radioactive sulfur in the supernatant. That's to tell you that in the presence of what? In the presence of virus, there's what? There's what? Sulfur. That's if you talk of what? Of this, because this is labeled here as the what? The protein code. And this is labeled as the DNA. So in this experiment, this scientist, Hershey and Chase, was now trying to what? To know that material. Before then, this protein code and DNA was not labeled. It was not known to be DNA and protein code. So they was now trying to know that substance that was able to transmit into the what? The virus, into the bacterium rather. They were now trying to know if this substance here was the DNA, this substance that was able to transfer here was the DNA or the what? The RNA. So when they now discovered that when they cultured it in the what? In two places, they cultured it in two places. They now injected what? Or put radioactive isotopes, which was sulfur and the what? The protein and the phosphorus. They were able to discover that in the supernatant there was radioactive sulfur. That's to tell you that in the supernatant there's the what? The protein code, because sulfur has to do with the protein, and in the pellets there was what? Phosphorus, and phosphorus has to do with what? DNA. So these scientists draw a conclusion that it's DNA that was able, this was still based on what? Avery, McLeod, and McCarthy experiment, which they said DNA was the what? Was the material for heredity. So this scientist here was now trying to what? To prove that experiment, Avery, McLeod, and McCarthy experiment, which they brought up this experiment as well. So they now concluded that in the presence of supernatant by centrifugation, the supernatant was containing radioactive sulfur, and the pellet was containing what? Radioactive phosphorus. So if in situations like this, if the supernatant was containing radioactive phosphorus and the pellet was containing radioactive sulfur, DNA will not be the genetic material today. It will be protein that will be the genetic material, because protein is found in what? Sulfur is found in what? Protein. Please, if you find this video confusing, comment in the comment section, like, share the video, and subscribe. Thank you.